Okay, we're gonna learn more about DNA today. So here, let's orient ourselves. This right up here, this is a strand of DNA. So we talked about the structure of DNA. So we can assume like these are all stuck together with our little hydrogen bonds. So both these sides are all stuck together. Here we have the original, two sides of the DNA. So we have a DNA strand, right? It's a ladder, remember? And it has the nitrogen bases in the middle and the sugar and phosphate on the sides. So that's our original strand of DNA. How DNA makes a copy. So we want to think back to an interphase. We had G1, where the DNA relaxed, and then in synthesis or S phase, it's when it made a copy. We're going to talk about that process. Like, how does DNA actually make a copy of itself? So here it's showing what it actually does. We see our old strand of DNA is the gray, and the new is the blue. So we call the gray the parent strand. Okay, and we call the new stuff a daughter strand. Okay, so we have the parent and we have the daughter. And what DNA does is it basically it pulls itself, it gets clipped in half, and then it makes the new copy on each side. So we call it semi-conservative. And in class we talked about conservative and dispersive, where different thoughts in the beginning of like how DNA might actually go through that synthesis of how it makes a copy of itself. But we know now that it's semi-conservative. So it takes one half of the DNA. Okay, so it takes that ladder and it cuts the ladder in half, and then it builds the other half, a new other half of the DNA. So today what we're going to be doing is we're going to learn about that process. So it's called DNA replication. To replicate means to make a copy of. So this is the synthesis step in interphase, okay? And it's when we're making a copy of the DNA. So here I'm just going through the slides that we did in class. So we want to orient ourselves in the beginning. So we have those two parent strands, just like we had up here, our two parent strands, okay? Here we're looking at it, a little section of it. Okay, so we've got two parent strands. We've got parent strand number 1 and parent strand number 2. Those end up later. One's going to be called the leading strand. Basically, it's one of the parent strands, it's leading, and one's called the lagging. If you think about the word lagging, it means like you're kind of behind, and it'll make sense later why we call it that lagging strand. So we have two strands, parent 1, parent 2, and one's going to be leading and one's going to be lagging. And we don't have to know which one's which quite yet. Okay, so our whole goal right now is to make that copy. So we're in synthesis phase of interphase. We need to take the DNA strand we have, and we need to take one strand and make it into two. So how we do it is we're going to take our parent strand and we're going to break it apart. So remember those hydrogen bonds that bind our nitrogen bases together? So if we look here, we have those hydrogen bonds that are holding them together. We can break those apart. And how we do it is we use an enzyme. So enzymes, remember, they speed up reactions and they can either break things apart or bind things together. Well, we are going to use an enzyme, and actually your body uses an enzyme to break it apart. So it's going to take your DNA strand that has the two halves of the ladder, and it's just going to zip it apart. It's going to break it apart. Okay, so this here is an enzyme, and the enzyme is just breaking all these bonds. So then these are not stuck to each other anymore. The A is not stuck to T, the G is not stuck to C anymore. So that's our first step. First step is we have to actually pull apart the DNA strand. Take the two sides of the ladder and break the rungs in the ladder. Okay, so we have different enzymes that are doing all these jobs. That one that breaks it apart is called helicase, and we know it's an enzyme because it ends in ase. And helicase basically is unzipping the DNA. The DNA has the two sides of the ladder stuck to each other. The helicase comes and it takes that ladder and it breaks it. So you don't have a very functional ladder anymore, right? You wouldn't want to use it anymore. It only has half of it. So we have two halves of the ladder. But helicase has to come in and it breaks the two strands apart. So we have parent 1 and parent 2 side of the DNA that are pulled apart from each other. Okay, so we're breaking those hydrogen bonds. So here's helicase doing it. We can see helicase here is breaking apart these hydrogen bonds that are holding the different nitrogen bases together. Okay, so now you've got this DNA, right? Here's our strand where it's all stuck together. We have our nitrogen bases that are held together with a hydrogen bond. But here, we still have our nitrogen bases, but helicase has come and it's broken those connections. Just imagine those all line up perfectly, which they don't right now, but that's okay. So helicase has come through and now we just have a parent strand here and a parent strand here that are loose. Our next thing that we have to do is, we need to build the other side of the ladder. So helicase came and pulled them apart. Now, something called DNA polymerase, again, it's another enzyme. We call it the builder. It's going to come in and it's going to attach new nucleotides. So remember, our nucleotide is made up of a phosphate, a sugar, and a nitrogen base. DNA polymerase is going to take nucleotides and attach them to the parent strands. And DNA polymerase does it with, we call it complementary base pairing. So complementary just means it's following Chargaff's rule. A goes with T, G goes with C. So here we've got our builder, okay? So think of him as Bob the Builder if we need to. They're going to come in and they're going to attach the complementary nucleotide. So with this, if we have a T, we know an A will get attached. We know if we have a C, a G will get attached. If we have an A T will get attached, and another T. If a T and A will get attached. If we have a C, a G will get attached. And it's not just the nitrogen base, it's the whole nucleotide, but the part that they're sticking together is a complementary one. Okay, so we call DNA polymerase, I call it the dumb builder. Mostly because it has no idea where to start. So it wants to build something, it just doesn't know where to start. So in DNA, there's something called a primase. So primase tells us it's the thing that it's the starter. It's like here we've got, if you're doing a race, it's the person with the flag telling you when to go. So primase kind of flags down DNA polymerase and is like, hey, you need to start here. So primase calls, it says, hey, come over here, start over here. So, and DNA polymerase will come and it'll start wherever that primase is. So again, primase just tells the DNA polymerase where to be. Like, where do I need to start? It doesn't know where to start. It needs to be told where to start, which is why we call it the dumb builder. Okay, so we've got all of these players that are doing their part. We've got our helicase, which is going to unzip the DNA strand. We're going to have the ladder's going to get split in half. And then the DNA polymerase wants to build the new strand attached. It wants to build the new part of the strand, but it doesn't know where to start. So primase is going to tell it where to start. And then DNA polymerase, the dumb builder, will to start building the strand. Okay, in the next video, we're going to talk about leading and lagging strands and Okazaki fragments and some more things that go on in DNA replication.